dive into our theme of today, that's the aging of the brain. Well, since that our audience are normal people but not professional researchers, then could you please briefly introduce the neurodegenerative diseases to us first, such as the Alzheimer's disease, Parkinson disease, then you may include the symptoms, risks, or vulnerable populations?
Sure, absolutely. Love to. So starting with Parkinson's, traditionally Parkinson's has been characterized by loss of voluntary motor movement. And that loss is due to death of a set of neurons in the midbrain in this region called the substantia nigra. And these neurons contain the neurotransmitter dopamine. And these cells are part of a larger neuronal circuitry which controls motor movement. So you have this sort of electrical circuitry, and when you lose these dopaminergic neurons in the substantia nigra, it's like you've cut through the wire and all the lights go off. It's interesting that this term substantia nigra is due to the fact that these neurons, which contain dopamine can be converted to a type of melanin called neuromelanin, and as a consequence, the neurons appear black in appearance on, postmortem autopsy. And if you compare the brain of a Parkinson's patient to that of an age-matched control, you can actually see the reduction in levels of these neurons. The disease also includes buildup of this protein called alpha-synuclein. And this was really discovered, wow, I can't believe it, but like over 20 years ago, where people discovered that there were some rare familial cases of Parkinson's disease. And when they isolated the mutation in this family, it turned out to be in a gene that encodes this alpha-synuclein protein. And then when people went to look at people who have what's called sporadic Parkinson's, where there's no genetic mutation or familial component, they found that within the neurons of even people who don't have an alpha-synuclein mutation, they have a buildup of this alpha-synuclein protein. And so over the course of the last couple of decades, that's been a major topic of interest for researchers in the Parkinson's field. Because you're affecting this voluntary motor movement, when you lose these neurons, patients experience symptoms like freezing or tremor, loss of balance. It's usually in sporadic cases, it's initiated like around 65 to 70 years of age. It's interesting, it tends to be more common in men than in women.
Yes, many of our audiences also reported that their parents are suffered from these diseases and also have these symptoms or indication.
Yeah, I mean, somewhat ironically, my mother, who's in her 80s, was diagnosed 3 or 4 years ago with Parkinson's disease. And as we may get into, she, like a fair number of Parkinson's patients, experienced gastrointestinal issues 10 to 20 years before she was diagnosed with Parkinson's. And that is a not uncommon feature of PD. So
sorry to hear that.
Yeah. So there in the last, a couple decades, there's been a lot of emphasis on looking at the genes involved in Parkinson's. Not that these gene mutations are responsible for many of the cases of PD, only maybe 10%. But the thought was that by understanding what if mutations in these genes are causing a problem, then it kind of points to particular pathways that maybe even when they go awry, play a role in sporadic cases of the disease. So there has really been, with both Parkinson's and Alzheimer's, a lot of emphasis on genetics. But for those of us studying aging, we realize that aging, genetics really only contributes to maybe 10% of aging. And so there's a growing recognition in aging itself and in age-related disease, a growing appreciation of the role of environment. And Parkinson's disease is something that's been known and studied for quite a while. There's a strong link with environmental factors, including exposure to certain pesticides like paraquat or rotenone, which is something which are used on to rid crops of what do I want to say, of things that would be harmful to them. But there is this very strong epidemiological connection between, for example, a farm worker's exposure to pesticide and later developing Parkinson's disease. In fact, it's probably one of the environmental toxins which has the strongest tie to a disease state. And that's led to use of toxins like paraquat in preclinical models to study Parkinson's disease. It's also, so there's this risk with the environment. I think in terms of Parkinson's disease, and I think we more and more think of this in terms of many, many different age-related diseases, but there's kind of this triple threat, which includes not just aging. A lot of these chronic diseases, the older you get, the more you're prone to present with these. There's also genetics, and it may not be a familial mutation, but there may be genetic predisposition. For example, with cardiovascular disease, people may have certain alleles or certain forms of genes that affect, lipid metabolism. So they're going to be more prone to having cardiovascular disease. And then if you couple this with no exercise, poor diet, stress, this all kind of works together to contribute. So it's kind of this triple risk factor: aging, genetics, and environment that we really think contribute to a lot of this.
As you have mentioned, that aging itself and aging-related diseases may play an important role in neurodegenerative diseases. Well, can we clearing the senescent cells using, for example, senolytics and other drugs can effectively prevent brain aging and other neurodegenerative diseases?
So senescence has been, I'll tell you a story. I was part of a grant from the NIH about 15 years ago that involved somebody who's very prominent in the senescence research field, Judy Campisi. And this grant was really looking at how the mitochondria, these power plants in the cells that produce needed energy for all of the cellular functions, we're really looking how mitochondrial dysfunction contributed to aging and various age-related diseases. And during the course of working with Judy as part of this grant, I sort of turned to her and said, it seems pretty clear that that cells in the periphery, when they undergo senescence, this can contribute in various tissues, contribute to disease, but it doesn't seem like many people are looking at the brain. The dogma at that time was that senescent cells had to be replication competent. And I think when people think about the brain, they usually think about neurons, which are post-mitotic. But in fact, the brain has a number of cells which support cells, glial cells, which are actually able to replicate and will replicate under stress. conditions. So interest over it's really just been in the last 5 years, interest has really become very fierce in looking at the role of senescence in Alzheimer's and Parkinson's disease. And in fact, as you and your audience may know, use of senolytics as a treatment for Alzheimer's is currently under clinical trials, including at Wake Forest. I would, based on our research, make a note of caution in that when you're giving senolytics to a multicellular organ, which most organs frankly are, there may be an issue with finding a dosage of the senolytic that's going to selectively remove the senescent cells without impacting on healthy cells. So in our laboratory, in long-term human brain cell cultures, our data in treating these cultures so first producing Alzheimer's in a dish in these cultures by giving them A beta oligomers. These cells, both the glial cells and the neurons, undergo senescence. And if we treat with a senolytic, we find that, for example, while cells like senescent glial cells, astrocytes, thymic glial cells, may respond to a particular dosage of a senolytic in the desired manner, you get selective killing of senescent glial cells and not other cell types, it may actually have detrimental effects on healthy cells like neurons and could kill those cells. So I think that's an important consideration. And one of the things that we're interested in our lab is developing more targeted approaches to administer drugs like senolytics using genetically engineered cells like immune cells. And the idea is that you can develop, so we're trying to develop systems, cellular delivery systems, where the immune cell will only, it will travel to a site of pathology, for example, in an Alzheimer's brain, like an A beta plaque. It is genetically engineered to produce drugs like senolytics that can prevent loss of, let's say, neurons. And then once the A beta pathology has been removed, then the system turns off. So I think we need to be really thinking about how we're going to target drugs, because right now, even for things like immunotherapy in Alzheimer's disease, you're giving an antibody against A beta. You have to give a really high dose antibody for it to cross into the brain. And it's not only impacting on the plaques and tangles, but because it's bulk drug delivery, it's going to impact on healthy cells as well. So that is, in fact, we just recently received an NIH transformative award grant, which is, these are kind of high risk, high reward to test these kind of cell-mediated drug delivery systems. So we're really excited about that.